All right, guys. Good evening, everybody. Welcome back again to another edition of the DY Patil Lecture Series. We are privileged to have you all with us, and even more privileged to have with us Dr. Wahiya this evening, who's been a teacher, a mentor, an inspiration for many of us, and he continues to be so. And we have, on his request, actually, this topic, epigenetics and psychiatry, and Dr. Ashutosh, who's now turning out to be a golden find, and he is who keeps laying our golden eggs, so we keep getting him back to our fold from time to time when we feel like having an academic omelette. And he is with us today. It is my privilege to introduce Dr. Wahiya. Dr. Wahiya is obviously, everybody knows him, so introducing him is like saying the sun is bright. So, but nonetheless, we have to follow protocol. Dr. Wahiya is an international distinguished fellow of the American Psychiatric Association. He is a professor emeritus of psychiatry at R.N. Cooper General Hospital and HBT Hindu Medical College at Mumbai. He is the founder and head of Department of Psychological Medicine at Cooper Hospital. He is formerly the professor of psychiatry at GS Medical College in Cooper Hospital. He is a consultant psychiatrist at Breach Candy at Leelavati Hospital. He is a member and National Advisory Board of the National Advisory Board of the Indian Association of Private Psychiatry, former international member of the American Psychiatric Association Membership Committee. He is a life fellow of various psychiatric bodies, including the Indian Psychiatric Society, the IAPP, the Psychopharmacology section of the IPS. He is the chairman of the CME committee at the IPS as well as the IAPP, and he is a member of the Positive Psychiatry section of the World Psychiatric Association. He is a founder, managing trustee, and CEO of the NS Wahiya Foundation. And he has several publications in index journals, medical journals, chapters in textbooks and monograms. And most importantly, he is an excellent human being, humble beyond one could possibly fathom. And he is now a part of a legacy of the Wahiya household. He is the second of the three generations of Wahiyas, and going by how strong the lineage is, we can expect many more future generations to contribute to the field of psychiatry. So welcome, Dr. Wahiya, and thank you for making the time. I know you had to shuffle your schedule to accommodate us, but we are forever grateful to you and constantly inspired by you. And I welcome you to take this evening forward, and I'll come back at the end to just give my concluding remarks. Thank you. Over to you, Dr. Wahiya.
Thank you, Ashwin. You're very generous by talking about me, and I'm indeed grateful for a whole lot of nice things that you have said about me. It's a pleasure to hear you talk about me, let's say. Why this topic of epigenetics? So, ever since I joined medical college in 1967, we were told that you are what you inherited from your parents. Apparently, that's not true. Individual characteristics that are intrinsically inherited from the parents is not just it. There is seemingly much more to it. There are extrinsic environmental factors which modify the genetic composition with which a person is born. Some of us, including me, of course, were taught that humans have 23 genes and all of them are diploid genes, meaning it has genetic material woven into two strings of nucleotides, and it's formed at fertilization from two haploid genes, X from the mother and either a Y from the father or another X from the father. This haploid genes are a part of the DNA, and as I understand, they reside in each nucleus of the cell. Things get more interesting. Humans have 3 billion nucleotide bases in their DNA. And the sequence of these nucleotides differs marginally in each individual, specific to each individual. And this is the basis of DNA profiling. Mother Nature didn't end here. There is more. It has ensured that the external and the internal environment have the power to regulate how genes express themselves. And this part of genetics is epigenetics. The word epi comes from the Greek, which means over or on top of genes. So epigenetics is something which is on top of the genes. And epigenetic is that additional layer that lies on top of the DNA and controls how the genes are read and how they express. We, as mental health professionals, got sensitized to this after the worldwide genome study or the international genome study, which gave not so reliable data. Because there is something called the heritability gap, meaning the heritability is studied on families of patients of schizophrenia, and what the gene study or genome study should reveal are not the same. So what is this heritability gap and why does it occur? And this is particularly important as we talk of, in the era of exon sequencing, etc. So why Ashutosh? Why not Ashutosh? Who else but Ashutosh? I've known Ashutosh since 1999 when he joined Cooper Hospital as a resident. He had experience of general medicine department working in private hospitals after his graduation from Terna. So he has been in medical field since 1992 and joined psychiatry in 1999. And after his DNB, he has had international exposure when he worked for a pharmaceutical company as a medical advisor, and he participated in several studies. After he came back to India, he's been with me, he's a part of my team at the Dr. H.D. Gandhi Hospital. And what I find about Ashutosh is his peculiar, very, very classical and not so common insight into the biological psychiatry. So he's a swan, if I may say so, unpatriotic towards psychotherapies and psychoanalysis in particular. But he is into biological psychiatry in a big way, and we'll soon find out. Dr. Ashutosh, will you please take over?
Thank you, sir. Thank you for the kind words. And thank you, Dr. Ashmin, for inviting me today. And thank you, everyone, for taking time out of your schedule to join us over here. I will start by sharing my screen. Why, sir? Let me know if you can see my screen, please.
I can, yes. I see a sky, which I believe is come from your screen. Yeah, now we see the PowerPoint also.
Right.
This PowerPoint or?
Keynote.
The keynote, yeah.
So, if you all have ever wondered how life evolves, sustains, and replicates, this presentation is going to give you some glimpse into this particular question. I'm Ashutosh, and I'll be speaking on epigenetics for beginners. And before I delve in, a little bit of warning that I'll be covering only the basics, and an immediate translation into your clinical practice is unlikely. However, it will be useful for the years to come. So moving ahead, the agenda, which I will take roughly around 60 minutes of your time, will be into 4 parts: 3 slides on evolution of life, 2 slides on self, an in-depth review of genetics, without which an understanding of epigenetics is going to be difficult. So starting with evolution of life, Earth is said to be around 4.5 billion years. That's billion years. And the climate of the Earth, the primitive Earth, was quite identical to the current day, except that there was a lack of molecular oxygen. Now, the primitive Earth had these elements: nitrogen, water, ammonia, methane, carbon dioxide, carbon monoxide, which were the raw materials for starting the chemistry of life. And the energy which was required to break the chemical bonds and also create new chemical bonds was essentially provided by the ultraviolet radiation from the sun. Remember, that time there was no oxygen in the atmosphere or any protective ozone layer around the earth. So all the ultraviolet radiation reached directly to the surface and on the sea. And that plus the electrical storms on the planet, along with the volcanic heat, gave the energy to form life. And this hypothesis was first tested in 1953. So almost 7 decades back by a scientist called Stanley Miller. You see him over here. And this is the apparatus in which he tested this. So what he did was he combined four things: water, ammonia, hydrogen, and the whole apparatus was using the boiling water for circulation and energy source by the electricity. And after a week, what he found was there were a lot of organic compounds. So when we say organic compounds, I take you all back to the time you were in high school. This is a compound which contains a carbon-carbon bond or a hydrogen-carbon bond. That's an organic compound. And that plus amino acids, which are the building blocks for proteins. And then subsequently, similar experiments reveal that other biological important molecules could be synthesized, because in this whole reaction, the first two chemicals to be manufactured were hydrogen cyanide and aldehydes. And then subsequently, the other important biological molecules could be synthesized. And of course, nitrogen bases, which are an important component of nucleic acids, as you will see in my topic on genetics, these were also produced from the hydrogen cyanide and ammonia. So having seen this, we have to understand that the first property of life is its ability to reproduce. And the molecular mechanism for reproduction is actually coded by the nucleic acid. And the first living organism was probably either a large nucleic acid or a polymer which contained nucleic acid. And this had an inherent ability to form a cell-like structure. And the organism depended on the soup of organic compounds. So just imagine this is happening in the sea, and around this particular nucleic acid, there are other organic compounds, and they are giving the nutrition to this particular nucleic acid, as well as other compounds to form its structure. So it was totally dependent on the soup of organic compounds. And once this organic compounds got used up, a new source of energy had to be found. And that is when photosynthesis said to be evolved. And what photosynthesis does is it captures the energy from the photons in the sunlight and stores it in the form of a high energy phosphate bond and adenosine triphosphate. And this is what would give energy when it gets converted to adenosine diphosphate. So one phosphate bond breaks. It is inherently quite an unstable molecule. When it breaks, it releases energy, and this energy is used to manufacture sugars from simple products like carbon dioxide from the air and hydrogen, which is got from the water. And the end product was oxygen gas. So remember, the primitive Earth did not have oxygen gas. It was photosynthesis which started producing oxygen gas. And this is what is hypothesized, that most of the oxygen in our atmosphere evolved or came from the photosynthetic reactions. And when enough oxygen had accumulated in the atmosphere, a more complex form of cell evolved, which used this oxygen to actually break down fats and sugar to provide energy. And then subsequently, this developed into a more complex organism, because using oxygen to break down fats and sugar is a more advanced and efficient way of providing energy. And then subsequently, roughly 560 million years back, there was an explosion of life forms, what we call the Cambrian explosion, which led to all the diversity of life that you see today. The other property of life is its organization. So the first property is self-reproduction, which is coded by nucleic acid. And the second property is organization. So any organism is a coordinated organization of what we call a structural unit, the basic unit of life, the cell. And the cells are largely composed of polymers, which have an inherent capacity to organize into a structure and replicate themselves. So polymers are actually what is, if you search online, you will find that polymers is having multiple subunits, and these are multiplied subunits of the same molecular structure. These polymers actually evolved from small organic molecules almost 4.5 billion years back from just a simple combination of four things: hydrogen, oxygen, nitrogen, and carbon. And it was actually the unique characteristic of carbon atoms to bond to each other to form a long stable chain, which actually made the evolution of life possible. So with this brief glimpse into evolution of life, I move on to the second part, which is the cell. And what we are now appreciating is that genetics is playing an increasingly important role in the practice of clinical medicine. And it is also playing a central component in our understanding of most medical illnesses, including psychiatric disorders. Genes, they affect each component of the human body. And as psychiatrists, we should understand the science of medical genetics. So this is a cell over here. What you see is known as a eukaryotic cell. Eukaryotes means they have a nucleus, vis-a-vis a prokaryote, which does not have a nucleus. So the human body is a social order of anywhere between 30 to 100 trillion cells. So billion is 9 zeros, trillion is 12 zeros. So you can imagine the quantum of cells that are there in our body, and these are organized into various functional structures, the largest of which we call an organ. So over here in this cell, we see there are many components, but before that, the basic constituents of the cell. A cell consists of around 70 to 85% of water. The next large component of cell, anywhere between 10 to 20%, is protein. So proteins, they provide two things for the cell. They give a structure. So just imagine if you have to erect a building, the building has a beam which supports the structure. Something similar, the structure of the cell is given by the proteins. And all the chemical reactions which happen in the cell, they need a catalyst, an enzyme. That enzyme or catalyst is the protein. So they have a functional role. And of course, a third role will be they have a pathway across the cell membranes and different membranes within the organelles of the cell. So they provide a pathway, a channel within the cells. So that is the function of proteins. Now, other than this, there are ions within the cell. These largely are magnesium, potassium, phosphates, bicarbonates, and sulfates, and some amount of sodium, chloride, and calcium. So these are also present in a cell. The next component, which constitutes 1 to 2% of an average cell, is lipids. So lipids, by example, are cholesterol, triglycerides, phospholipids. These are the energy storehouse, as well as they form constituents of the cell membrane. And then the final thing is carbohydrates, which is usually 1%, and they are stored in the form of insoluble polymer of glycogen. But in cells of the muscle, they can be as high as 3%, and in the liver, 6%. So this is the chemical constituent of the cell. The cell has a cell membrane, which kind of separates the contents of the cell from the interstitial fluid. And then various organelles. This is the mitochondrion, which gives energy. So it provides the energy to the cell. This over here is the endoplasmic reticulum, the Golgi apparatus. These are the sites, specifically the endoplasmic reticulum, which manufactures the lipids, the proteins, carbohydrates, even certain structures like lysosome or peroxisome. And then the Golgi apparatus, this further processes the proteins which are manufactured by the rough endoplasmic reticulum. So the ribosome bounds to this particular endoplasmic reticulum membrane to form the proteins. And we'll see in the genetics how the proteins are actually formed. This structure over here is the nucleus. and this has a nuclear membrane, and there are nuclear pores from which the RNA can come out of the cell. And here you have the chromosomes, which Dr. Vaya had explained to you all, that each human body has a diploid set of chromosomes, 23 diploid chromosomes, one from the father, one set from the mother. And then you have this nucleolus. This is nothing, this does not have any membrane. It contains enzymes and proteins as well as the ribosomal RNA. And this is then excreted from this particular nuclear membrane and forms the ribosomes in the cytoplasm. Now, it is estimated that in the human body, different cell types produce over 100,000, that means almost 1 lakh plus proteins within the different cells of the body. So it's a huge number of proteins. And on an average, so Dr. Vaya did tell you all that there are 3 billion base pairs of nucleotides, which have a coding for roughly 20 to 25,000 protein coding genes or exons. We'll see about this later. So this was the basic about the cell, and I move on into genetics. So first of all, the nuclein or nucleic acid was actually identified by a Swiss physiological chemist called Friedrich Mescher way back in 1860s, 1869 to be precise. So it's almost 150 years plus. This particular scientist identified nucleic acid. He called it nuclein, Nuclein, nuclein. And it was never kind of known for many decades. Subsequent scientists, chiefly Phobos Levin and Erwin Chargaff, they deciphered the chemical structure of this nucleic acid, including how it is organized. And without this particular, I would say, groundwork, it would not have been possible for Watson and Crick in 1953 to come out with the groundbreaking paper. It was actually just a one-page paper for showing the DNA structure organized as a double helix, which got them the Nobel Prize. So Watson and Crick did not actually discover the DNA. They described the structure of DNA as a double helix structure. And this is just a repetition of what Dr. Vaya said. Each human genome has 23 chromosomes, numbered 1 to 22 as autosomes and one sex chromosome, either X or Y. And over here, you will see the blue one, which indicates from the father, the paternal, and the pink one from the mother. And the somatic cells are diploid. So they have 23 chromosomes. And Y chromosome is necessarily coming from the father. And the mitochondrial genome, this is solely derived from the mitochondria in the ova, an exclusive matrilineal inheritance. However, it's becoming controversial in the last few years, where also there is evidence being proposed that it could be also coming from the paternal side, but I will leave that out. This is the actual photographs of the chromosomes, and you'll see these bands. That's the actual genes which are there, the protein coding genes. And the long arm of these chromosomes is given a alphabet, which is called T for the long arm and Q for the short arm. So whenever you read something like 11P, whatever, it means chromosome 11. P stands for the long arm, and then the subunit, etc., which gives the precise location of which gene is somebody talking about. So having seen this, this is a pie chart showing the structural composition of the human genome. And actually, this particular intron-exon structure was kind of discovered in 1977 by two scientists, Phil Sharp and Robert Richard, which got them the Nobel Prize in 1993. And it is this 1 to 2%. You see such a small component of what is known as the protein coding exons, which actually is there in our genetic structure. The remaining largely is introns. These are the non-protein coding. And obvious question is, why so much of introns? So the hypothesis is, by lengthening the length of the chromosome, these introns, they facilitate a shuffling of the different genes during the process of meiosis. And the other thing is, by lengthening or varying or modulating the time required for DNA replication, and transcription. That is what is the hypothesized function of the introns. And of course, now we also know that this is not just an inert part of the chromosome. It also plays a good amount of role in the epigenetics. These other structures, I will leave them out of this basic presentation. And this is just a representation of how a typical chromosome would look like. So you have a regulatory region, which would regulate, as the name suggests, and the gene, which is the protein coding component over here, which actually contains the genetic code for manufacturing different proteins. And then, of course, the intergenic regions, which separate different genes from each other. So let's look at DNA. So DNA stands for deoxynucleic acid, and it consists of three things. A phosphoric acid over here, you see this triphosphate, a sugar, which in the case of a DNA is a deoxyribose. Why is it called deoxy? Because in the RNA, over here I have circled in red, it's small, so a little bit strained for seeing this. This has a hydroxyl group, and the oxygen is missing. That's why a deoxyribose is just a hydrogen bond over here. So there is a phosphate, there is a sugar, and there is a nitrogenous base. So this is the primary component. So just a phosphate and a sugar is what is known as a nucleoside, SIDE. And when you have the phosphate, the sugar, and the nitrogenous base, it's called a nucleotide. And the human genome consists of 3 billion, B as in B, nucleotides overall. And you have two types of nitrogenous bases over here represented, the purines, which have actually 2 carbon rings over here, you can see. One is the adenine, the other is the guanine. And then pyrimidines, which have a single cyclic carbon ring over here. One is thymine, the second is cytosine. And they are represented by the alphabet CTAG. C stands for cytosine, T for thymidine, A for adenine, and G for guanine. So this is the basic of DNA, and this is a double helix structure. And of course, I won't go into details, but this is a minor groove, major groove. You have different kinds of DNA, A DNA, B DNA, Z DNA, depending on if it's a right-handed, left-handed, etc. But that's out of the basic thing. So just for your information, there exists multiple types. The most common is the B DNA. Now, this is a little bit more details. So you will see over here the 5' and why the 5'? Because if you see the previous slide, this is the numbering of the carbon. This is carbon 1', 2', 3', 4', and 5'. So therefore, the 5' end of one strand is paired with the 3' end of the other strand. So it's called an anti-parallel structure. And then, of course, the cytosine and guanine, they pair with each other. So cytosine over here is a pyrimidine, which pairs with the guanine, the purine over here. And that pairing is happening through these hydrogen bonds. So there are 3 hydrogen bonds over here. Same way, adenine with thymine with 2 hydrogen bonds. And these are relatively weak bonds. Of course, you will also see there will be a possibility for this hydrogen over here to bind with something else. And that is where the methylation actually happens. So it's possible that another methyl group can be added over here without affecting the structural integrity of this particular DNA double helix. So this is a little bit in-depth view of the DNA molecule. And this is known as the backbone of phosphate and sugar. So there is alternating phosphate, sugar, sugar, phosphate. So this forms the backbone, and this are having the hydrogen bonds. And the 5' end of one sugar bonds with the 3' end of the other phosphate, and it continues that way. So this is an overview of the DNA. Now, if we actually were to put DNA as a single line, it would be as long as 2 meters. Now you imagine this 2 meters of DNA has to be fitted into a size of nanometers. This requires coiling. So this coiling is what is shown over here. There is a histone core. So histones are protein, and these, I will explain the components of the histone in the coming slides. But roughly 140 to 150 base pairs of these nucleotides are wound around this histone core. And then you have around 20 to 60 spacer elements that comprises of 20 to 60 nucleotides. And then these 6 of these, what is known as a nucleosome, they form a solenoid. So there is kind of curling around, turning around, and it kind of compresses the whole structure. Following the solenoid, there is a chromatin loop which consists of roughly 100,000 base pairs. So 1 lakh base pairs. And then subsequently, it's still not known how it is further condensed. But the end result is it is compressed to 1/10,000 of its size. And that is how this 2-meter long DNA is actually fitted into the cell. And this structure is what is really important, and I will explain to you how. So the next slide is actually showing you the structure of known as chromatin. So chromatin is nothing but a DNA mixed with RNA along with protein complex that is known as the chromatin. And this is the histone core over here. You will see this is the histone core, around which this is the nucleotides which are bound. Roughly 140 to 150 base pairs are bound around one histone core. Now, when the histone core is tightly packed, so you imagine there is no space between the two nucleosomes, that is known as the heterochromatin. At that point in time, it's not possible to read the contents of the nucleotides. So the gene is said to be switched off. And of course, the reverse, when there is a good amount of space, the genes can be read. It is known as the euchromatin. And of course, these cores consist of 5 classes of histone. So you see over here the H1 class, and then you have an octamer. Octamer means 8 units, which have 2 units each of H1, H2, sorry, H2A, H2B, H3, and H4. So that is the component of the histone core. What is important to notice, these histones are positively charged. Why? Because they have at least 20% or more of these basic amino acids, lysine and arginine. So what does this positive charge do? If you remember the structure of DNA, it had phosphoric acid, which is negatively charged. So if you have to compact this particular DNA, the negative charge would repel. So by kind of eliminating the negative charge, because the histone core is positively charged, it reduces the electrostatic repulsion and permits the DNA to be compressed and packed tightly inside the cell. And that is what is depicted over here. So in this particular diagram, you see the histones are loosely packed. So something can come and read what is written on the genetic code and process it further. So transcription is possible, or what we say the gene is switched on. And the opposite, when it is tightly compacted, it is not easy for something to come in and read what is written, and then thereafter transcription is not possible. So the gene is said to be switched off. So this is known as a euchromatin, and this is known as a heterochromatin. We'll touch base on this ahead in the presentation. So having seen this, what is important is one slide on DNA replication. So we are all born as a single cell structure. Our life started as a zygote, and then we became a miraculous structure of 30 to 100 trillion cells, essentially by the process of DNA replication. But other than this, throughout our life, our cells do not last the full life. They have to be continuously replaced, new ones generated. And the key to this identical replication is something because of complementary base pairing. That means adenine has to be always paired with thymine and guanine with cytosine. This consistency is what makes a DNA replication accurate. And the main enzyme involved, this one over here, you see the pinkish one, is known as the DNA polymerase. And if you remember, the pair, the DNA 2 strands are just attached by a weak hydrogen bond. So first, something has to unwind. So break this hydrogen bond, unwind the 2 strands. Then second thing has to hold the strands apart. And then this DNA polymerase is going to add the new nucleotides. And that it does from the 5' to the 3' end. So remember the 5' was the carbon, where the carbon is, and the 3', the 2 ends of the particular anti-parallel structure. So 5' end is known as the upstream and 3' is known as the downstream. And the DNA polymerase can add new nucleotides only to the 3' end. It cannot add to the 5'. So it starts over here and it keeps on adding. And this is the new DNA strand in orange, which you see here. And that is how the DNA replicates. And of course, the DNA polymerase also does part of what is known as a proofreading. So it has to check that this adenine has been paired with thymine and guanine with cytosine. Of course, in the case of an DNA, this is what happens, but it has to ensure that this is indeed the case. If a wrong nucleotide is added, it has to excise it and replace it with a new nucleotide. And if this process does not happen, then a mutation is said to have occurred. So this is how a mutation would happen. This is about DNA replication. And what is important to remember is, in the humans, the rate of DNA replication is relatively slow, almost 10 to 20 times as slow as that in a bacteria. So of course, if a gene in the humans has 250 million bases, it would take almost 2 months for it to replicate. Very slow. So in order to speed up the whole thing, replication begins at different sites called the replication origin. And then it forms a replication bubble, and eventually the two daughter strands, they separate out. And this is how the whole replication process is speeded up in the human beings. This is a general scheme which shows how the genes control the manufacture of proteins. So you have the DNA over here, there is a transcription. So something reads what is written and it transcribes into a RNA, a ribonucleic acid. And this RNA leaves the nuclear membrane, comes out into the cytoplasm, where its instructions are read, that means translated, and something, and then it forms a protein. So that is depicted over here from the gene. RNA is formed by the process of transcription, and this RNA comes out into the cytoplasm. By the process of translation, it forms a protein. And the protein, as we already saw before in the cell part, serves two things. It gives us structure and stability. Second, it participates in the various reactions, chemical reactions in the cell as enzymes or catalysts, and therefore eventually controlling the cell function. So this is how genes control the cell function. And this second diagram is a little bit more informative. There's a process of transcription in the multicellular eukaryotes, followed by processing. Then this processed RNA comes out, is translated into proteins. So we begin by seeing how transcription starts within the nucleus. So now transcription is the process by which the RNA sequence is going to be formed from the DNA sequence. And the RNA which is going to be produced is called the M standing for messenger RNA. So it contains the instruction for manufacturing the protein. So this is the parent strand and this is the messenger RNA strand, and here the enzyme RNA polymerase. So to initiate the transcription, RNA polymerase is one of the enzyme which binds to something known as a promoter site. So promoter is a nucleotide sequence. Nucleotide, we already saw, consists of a sugar, a phosphate, and one of the nitrogenous bases. So there is something known as a promoter sequence which lies upstream. Upstream means on the 5' end, and that specifies to the RNA polymerase that it should start by transcribing from this particular point. Of course, RNA polymerase is not the only one. There is a host of other proteins which are involved. Something on the same strand which is to be transcribed, that is known as the cis response elements. And this consists of three things. One, the promoter. Two, the enhancer. And three, the response elements. We'll have a little bit details of this further in the presentation. But this is to tell you there's something known as a cis response element. And there is something, a protein which comes and recognizes, so it comes and attaches, that is a trans-acting factor, which is manufactured by another gene. So a trans-acting factor which comes and attaches to this cis response, cis-acting elements. I said response elements. These response elements, you can have different kinds, a steroid response elements, or you may have a response element to what is known as the CREB, the CREB, that's the response element. Something comes and binds, and that activates the process of transcription. So what does DNA polymerase do is, it pulls apart the two DNA strands and it attaches to one. Now, where does it attach? That depends on essentially the promoter sequence. So that either of these two strands can actually serve as the template for replicating into an mRNA. And RNA can be synthesized only from the 5' to the 3'. So the promoter specifies the directionality, which strand is to be chosen. So the strand which is chosen is called the anti-sense strand, and the opposite is known as the sense strand. So it kind of gives a complementary base pairing over here, and this is the new mRNA strand. Only difference is for RNA, the thymine is substituted by uracil. Otherwise, the whole complementary base pairing of cytosine with guanine is respected. And this new mRNA strand is identical to this particular sense strand. And the RNA polymerase moves from the 3' to the 5' end of this antisense strand, adding nucleotides to the 3' end of the mRNA. So this is the mRNA transcript which is getting generated by this RNA polymerase. Now, this complementary base pairing I already told you all, and the substitution of uracil for thymine. Now, this strand, the 5' end, is going to be capped by a chemically modified guanine nucleotide. Why? To prevent the degradation. And when this mRNA goes into the cytoplasm, it will be identified, this particular cap, and that will say, okay, start the process of translation from this particular site. Now the transcription will continue until something known as a termination sequence is reached. So once the termination sequence is reached, thereafter, there is something known as a poly A tail. A stands for adenine. So roughly 100 to 200 adenine bases are going to be added to this particular mRNA strand. And thereafter, that serves to stabilize this particular mRNA strand. And the transcription continues for a few thousand nucleotides even thereafter, but that all part will be lost. It will be completely cut out. So then what happens once the termination sequence is reached, the poly A tail is kind of put to this mRNA strand. Then the DNA strands, the RNA polymerase, and this new mRNA strand, they all separate. And then this is known as a primary transcript. Now you have something known as a housekeeping gene. That means
Ashutosh, there's one important question that has come up at this point, which says Shalini wants to know whether this is also known as telomeres.
no, this is not telomeres.
Yeah.
And I will take the questions right at the end.
Yeah, but this
Otherwise,
was one particular, this point I thought was important. That's why
no, telomeres are completely different. Telomeres
Right.
are something at the end of the chromosomes to prevent the
That's fine.
degradation.
Okay, I'm sorry to interrupt.
No worries.
Okay.
So the housekeeping genes, they are encoding the essential products required for maintaining and producing or keeping the cell alive. So they are transcribed in all cells of the body. All the 30 to 100 trillions of the cell, housekeeping genes will be transcribed. But most genes are transcribed only in specific tissues and at specific points in time. So just a small fraction of genes are actually actively transcribed. And this should help explain why each cell is so different. It makes such different varieties of protein, even though the genome sequence is identical in all the cells of our body. So many proteins participate in the process of transcription. As I said, you have the cis-acting elements, and then you have the trans-acting factors. So these trans-acting factors, you have something known as general transcription factors, which are required for all processes of transcription. And then you have specific transcription factors, which are required for very specialized role only for certain genes and only for certain stages of development. And this is a diagrammatic representation. So you have a coding region, you have the promoter region. Promoter, there is something known as Tata box. It's not the company Tata, but Tata stands for thymine adenine. And there's a thymine adenine replicates of around 100 to 300 of these pairs, and that forms a Tata box. So it's a recognition site. It tells this particular complex, okay, you start transcription from here, and the coding region is going to start afterwards. So this is the main enzyme involved, RNA polymerase, but it's not the only enzyme involved in transcription. Why? By itself, this RNA polymerase is not able to recognize this particular promoter sequence. So first of all, this RNA polymerase has to identify where is the promoter sequence. It cannot do that on its own. It needs this general transcription factors or basal transcription factors. A, B, F, E, H. These are different proteins. Plus, by itself, RNA polymerase is incapable of producing sufficient quantities of the mRNA. So therefore, if you remember the cis elements on this, along with the promoter, you have the enhancer. So enhancers, what they do is they enhance the process of transcription. So when the response elements as well as the enhancers, when they are acted upon by this activator transacting factor, so something comes, attaches to the enhancer, then this enhancer indirectly talks with this particular promoter by the activator factor, which in turn is attached to the co-activating factor, which in turn is attached to this particular tata binding protein and the complex, the general transcription factor, and then the RNA polymerase. So almost around 50 plus different proteins are involved in the process of transcription. And by this particular chain from the enhancer to the activator, to the co-activator, to the general transcription factor, very specific genes can be activated at different points in time. And of course, like enhancers, you also have silencers, which would do the opposite. They would silence the whole process. And these roughly around 1500 to 1800 plus transcription factors are already known. But that is out of scope for this presentation, which is just a basic view. So this is the whole process of transcription, which forms the what we call the primary transcript. And one more slide on the transcription factor. How exactly will that transcription factor know where the promoter region is? That is by this DNA binding motif. And you have different ones shown on the top of the slide. So what happens is this fits snugly and stably into the space. And then it is able to know, okay, this is the DNA sequence, which is the promoter region. So here shown in this particular bottom left is the helix-turn helix. You have different zinc finger. Many of you would have heard the BRCA gene for breast cancer. That's a zinc finger DNA binding motif. And there is a special kind of a DNA binding helix, which is known as the high mobility group, HMG, which can actually bend the enhancer from a nucleotide which lies thousands of base pairs upstream to the protein coding part. So all this is quite a complicated mechanism, and any mutation or any error in either the enhancer gene, the activator gene, the co-activator gene, the general transcription factor, or these DNA binding factors over here, that would lead to a genetic disease. Now, before this, what we call the primary transcript leaves the nucleus, it is further processed. So you have, by the process of transcription from the DNA, a primary RNA transcript, which is over here. And then you have certain enzymes which would remove these introns, which are the non-protein coding part. They would be kind of excised, and just the exons, these are the ones which give the code or the instructions to manufacture the different proteins. They are spliced as in joined. So imagine, forget if this is not the RNA. Imagine this is like a film the director has shot, and the editor is having different sequences. He has to cut and he has to paste them. And these exons can be joined in different ways. Over here, suppose we give this 1, 2, 3. This will code for one particular protein. But instead of this 3 over here, if it is 1, 3, 2, it will code for another protein. So one gene can code for more than one proteins. And that is the whole beauty by just 20 to 25,000 protein coding genes. And we have more than 100,000 different proteins being manufactured. This is the reason that the splicing which can happen can be shuffled. So there are different products from the same genes which can be manufactured. And here you see the modified guanine cap and the poly A tail that prevents the degradation of what is now called the mature RNA transcript. And this mature RNA transcript has to come out through the nuclear pores into the cytoplasm. And of course, I'm not going to the details as to how that happens, but if it is blocked, then the further processing and formation of proteins will be prevented. So this slide shows the genetic code. Now, what is the genetic code? So you remember that the DNA structure, there is possible of 4 different nucleotides, because there are 4 different nucleotides And if you remember your first year MBBS biochemistry, we have 20 essential amino acids. And all our proteins are actually polypeptides. And polypeptides, the basic structure is actually amino acids. So for 20 amino acids, if you have just 4 nucleotides, one nucleotide cannot code for these 20 amino acids, or even 4. Even 2 of these will not be able to, because then the permutation combination allows only 16 possible permutation combination. But if you have 3 nucleotides, then you have 64 possible different permutation combinations, much more than enough to code for all the 20 different amino acids. And this has actually been tested in the lab, where they synthesized the nucleotides and allowed them to form the different polypeptides. So this particular 3 nucleotide sequence, which codes for one amino acid, that is known as the genetic code. So over here, if you want to read, say U, U, and U, so 3 U nucleotides would code for a phenylalanine. So that is what is the genetic code. Now the genetic code, phenylalanine, can also be coded by U, U, and C. So that is uracil, uracil, and cytosine. If these 3 are there, it will also form phenylalanine. So one amino acid can be coded by more than one different codon. So the DNA or the genetic code is said to be degenerate. But one codon will always code for one amino acid. And if you are very careful enough to observe, 2 amino acids, methionine as well as tryptophan, there is only one codon which they have. They do not have more than one codon. And there are specific stop codons. Over here, UAG, UGA, and UAA, they will actually say stop. So no more protein production. That is a stop codon. And this is the genetic code slide which I have shown you. Now, the next process is translation. So we had seen that the mRNA primary transcript, which is formed into a matured transcript, which is then kind of comes out into the cytoplasm, where it is translated. That means the instructions in the messenger RNA would give a template for synthesis of a polypeptide. Now, mRNA by itself cannot bind to the amino acid. It needs to interact with this transfer RNA or tRNA. This structure over here is a tRNA or a transfer RNA, which has around 80 nucleotides, and it has 2 ends. A anticodon, which reads the codon over here. So it pairs. So if it's AUC, you have UAG, because that is how this will pair. And the other side, which contains an amino acid. So this 2 will be responsible for interacting. And the ribosomal RNA, the ribosome is the site where all this tRNA and mRNA bind to each other. And then this helps to form the polypeptide chain. So this is the ribosome over here, the messenger RNA, and these are the tRNAs which keep coming with different amino acids. Depending on which is the genetic code, the said amino acid, and this is the polypeptide chain getting formed. And the ribosome consists of enzymes as well as the ribosomal RNA, which comes from the nucleolus. So the RNA actually helps to bind the messenger RNA and the tRNA to the ribosome. So this polypeptide chain is getting formed. So this is the beginning of the process of formation of proteins. In the eukaryotes, before the protein is fully formed, it undergoes something known as a post-translational modification. So either the polypeptide chain is cut into small parts, or large parts, more polypeptide chains are joined to each other, or a carboxyl group is added to it. All in all, this is the protein structure. So you have the primary structure, secondary, tertiary, and quaternary. This is the stable protein. So in order to achieve this quaternary stable structure, the different parts of the polypeptide chains undergo further modification, that is known as a post-translational modification. And there is one example shown over here in this slide, is the formation of the collagen 1. Over here, you see from the nucleus, there is synthesis of pro-alpha chain, which is hydroxylized and glycosylated to form the triple helix, which is excreted out of the cell to form the pro-collagen, and then further cleaved to form the collagen, which assemble into the collagen fibril. So this is the last step of protein formation. You started with transcription, where you had a primary transcript, then the mature transcript. This mature transcript is sent out of the nucleus through the nuclear pore. Then the messenger RNA and the transfer RNA, they bind to each other to form the polypeptide chain on the ribosome. And then this particular protein or polypeptide chain undergoes post-translational modification to finally give rise to a mature protein product. So this slide is a FYI, for your information slide, to tell you that there are various classes of RNA, some of which are not actually involved in this process of manufacturing proteins, but they serve some other purposes. But I'm not going to go into details. But this whole kind of transform from genes to transcription to translation and then to protein, this entire process which is happening, there is a specific name for this in molecular biology and in genetics. So the gene expression, we saw that proteins are fundamental for functioning of the cell. They serve to form the enzymes which catalyze the chemical reactions. They give the structure to the cell. Genes control the protein synthesis and therefore the functioning of the cell. And gene itself is a double-stranded DNA molecule that controls the formation of RNA. And RNA spreads in the cell to form the specific protein. So gene expression is all this particular processes, right from transcription of the genetic code to translation and formation of proteins, that is known as a gene expression. And anywhere of the process, each has a specific control, each can be blocked. So there are multiple checks at each stages and multiple places where the process can be actually blocked. That is the whole point of gene expression. So the cell cycle, as I already said, our cells do not last a lifetime. They have to be replaced. And the process is by mitosis, that means a nuclear division, and cytokinesis, that is cytoplasmic division. And this, the cell duplicates the DNA during the interphase. So this, I won't be going into details. Over here is the gap one, wherein there is formation of the RNA and the different proteins. The S phase or the synthesis phase, where the DNA is actually duplicated. In gap 2, this duplicated DNA is kind of, if there are any errors, it is repaired. And then subsequently, the cell divides. And this alternation of mitosis with interphase is called a cell cycle. And the length of the cell cycle varies depending on the different tissues. What is important to note as psychiatrists is the neurons in the adults largely lose this ability to replicate. That means most of the neurons in our brain are actually existing in a post-mitotic state. So that completes about cell cycle. And with this, I will be moving on to epigenetics. So by now, you would have a fair idea of how genetics is there. We come to epigenetics. So for epigenetics, if each cell has an identical genome sequence, why is it that we have so many different cell types? The answer is because the chemical modification to the genome sequence. If you remember in that DNA structure, I told you all the hydrogen bond on the cytosine, the 5' site, is available for addition of different group called the methyl group. That is a chemical modification, as well as a chemical modification to the proteins that package this genome. So that's the histones. And this together is called the epigenome. So the different chemical modification of the genome sequence, as well as for the histone sequence, this will be the epigenome. So while the genome is identical for each cell, the epigenome is not similar between two different cell types, and even within the same cell at different time points and in response to the environment. And that explains why we have so many different cell types, despite the fact that each cell has the same genome. Now, a little bit of glimpse into history. Actually, epigenetics is not new. It goes way back into 300 BC, where it was called epigenesis. It was actually got to do with the formation of embryo. And it was the Greek philosopher Aristotle who actually first said that the embryo develops from an amorphous point. That time, the prevailing theory was the embryo comes from a pre-formation. So it's just a miniature version which grows up into a bigger baby. But that was not what Aristotle thought. He said it comes from an amorphous starting point and then that replicates into different structures. So actually the founder was Aristotle way back in 380 to 320 BC. Subsequently, in the 1940s, the British biologist, developmental biologist Conrad Waddington largely and Ernst Haddon, they actually understood that genetics and developmental biology were related. And we largely credit Waddington as the founder of epigenetics. In 1942, he published a paper where actually he combined the word epigenesis, that was to do with embryonic development, and genetics. And that is how the word epigenetics came into origin. And as per Waddington, epigenetics is the branch of biology which studies the interaction, causal interaction between genes and the products, which actually bring in the phenotype. And we define, of course, there are many definitions for epigenetic. I've given one definition over here. It's a modification of the genome heritable during cell division that do not change the DNA sequence. So that is epigenetics for your information. Now, the main primary components, as I said, is the chemical modification of the individual nucleotides that make the genome. And this is essentially by the process of methylation, and the chemical modification of histones that package the genome. And there are a wide variety of histone modifications that can happen. But together, these two are known as the epigenetic marks. So whenever you hear this word epigenetic marks, it actually stands for this, the chemical modification of the nucleotides that make the genome in that particular cell at that point in time in response to a particular environment, and a chemical modification of histones that package this particular genome. Now, nucleotide modification is largely methylation and hydroxymethylation. I'll explain these in the coming slides. And a lot of different kinds of histone modifications. Essentially, all the 4 different histones can be modified. The most common for neurobehavioral disorders, and the first one to be described, was acetylation. What is important to note is a direct causal relationship between the epigenetic marks and the genome functioning is still not well established. Research is still going on into this area. But what is also to be known for you all is, apart from these 2 primary mechanisms, there's something known as non-coding RNAs, which can also cause problems or can control this process of transcription, of translation, and protein post-translation modifications. But for this basic presentation, I'm going to leave that out for y'all to simply understand these 2 basic components. So coming to the nucleotide modification, so cytosine methylation. So this essentially happens when the cytosine is followed by a guanine in the genome. So if you remember the backbone structure, you have the different nucleotides, the nitrogenous bases attached by the hydrogen bond. If a guanine is followed by a cytosine, then the methylation is possible for the cytosine, and that is represented as a CpG. P stands for the phosphodiester bond between the adjacent dinucleotide. So if you remember the alternating phosphate sugar, that P stands for the diester bond between the phosphate sugar. And 5-methylcytosine was actually discovered in a condensed state of the nucleosome, the heterochromatin state. And that is why it was thought that it is associated with silencing the gene expression. So if you remember, in the heterochromatin state, the gene transcription is not possible because it is tightly, compactly packed. And therefore, 5-hydroxymethylation is thought to silence or prevent the gene from transcribing. And how exactly does this silencing happen? So 2 broad mechanisms. A, direct interference in the binding of transcription factor. So if you remember, this transcription factors, they come and they recognize the promoter sequence. So imagine in this tightly, compactly packed DNA, if there is no space for this particular molecule to come and bind, then there is no transcription which is going to start. So that's a direct interference, because now that hydrogen is replaced by the methyl group CH3. So you imagine this chemical is actually bigger. It won't allow this particular transcription factor to bind. So that's a direct mechanism. The other thing what happens is, once the methylated DNA is there, it recruits other factors which kind of cause the chromatin inactivating complexes. There is histone deacetylation, etc. So the histones also then undergo modification. Now, this is a schematic diagram which I have shown. So methylation, over here you see the CH3, that's the methyl group, and S-adenosyl methionine or SAM is the methyl donor. And the enzyme which causes this is known as the DNA methyl transferase, DNMT. DNA methyl transferase. Essentially, there are three families, DNMT1 and DNMT3, and two subfamilies of DNMT3, 3A and 3B. These 3A and 3B cause de novo methylation on one strand. The cytosine is methylated, and then on the opposite side, it is hypothesized that DNMT1 is the one which causes the cytosine methylation on the other strand. So we know exactly how DNA methylation is happening. The reverse, demethylation, is not well known. There are various proposed mechanisms. One is a passive replication, and then eventually the methyl group is lost. It's very energetically intensive. You just can't cut this methyl group. It requires a lot of energy, so it cannot directly remove that. And there are various enzymes involved. The TET is 1011 translocase, TET enzyme, then TET with TDG. So this is known as the eraser. This is known as the writer. And then you have something as a reader. One example is the MECP2. And I will show this in the few examples of the nucleotide modifications presenting with the different genetic illnesses. Coming to histone modification. So now we are shifting to the second modification, which is on the histone. So histone, just to recap, is an octamer. That means 8 units, which has 2 units each of histone H2A, H2B, H3, and H4. And there are different enzymes: histone methylase, demethylase, acetylase, deacetylase. And usually any of these can undergo modification, but usually the H3 histone undergoes either addition of 1 to 3 methyl groups or addition of an acetyl group. And you have different amino acids which can be having different modifications. So you may have lysine, which has methyl groups added. You may have lysine, which is having acetylation. You may have serine, either sumolation or ubiquitination. Different kind of chemical modifications can happen. And the most well understood is actually acetylation and methylation of the lysine residue. Now, in general, what happens is, once the acetylation of, let's say, the histone tail happens, it reduces the positive charge of this particular histone structure. So if you recollect, histones are positively charged. And what they do is they negate or they neutralize the negative charge of the different phosphate groups in the DNA and allow it to be tightly packed. Now, if acetylation is going to make the histone lose its positive charge, then this tight, compactly packed structure, it's going to repel. So it's going to be then loosely packed. That is how acetylation brings about the euchromatin state and a possibility of transcription. So this is how actually the acetylation of histone would enable the gene transcription to become possible, and the gene is said to be switched on. And the opposite, deacetylation, that's achieved by the histone deacetylase, or the short form HDAC. And just for information, you have 4 different groups of HDACs. Valproic acid, which we are all so commonly using, an anti-epileptic drug, a drug used in mania. It's a class 1 and class 2 HDAC inhibitor. So it can actually cause histone deacetylation and cause suppression of the transcription. So the genes can be switched off by using valproic acid because it's a HDAC inhibitor. So different, over here we see the different histone proteins and the different modifications and causing either repression or activation. I won't go into this details, but just to tell you all, if you read a code H3K27AC, you should not get flummoxed. What this shorthand notation, how do we read it? H3 stands for the histone. So by now you know there are 4 core histones, either H2A, H2B, H3 or H4. So this H3, K stands for the short form lysine, and 27 is the lysine on position 27. AC stands for acetylation. So this would be read as the histone 3, lysine 27 is acetylated. Same way, H3K4Me3 would read as histone 3. The lysine on position 4 has 3 methyl groups to it. And this is what you should be able to read whenever you read any paper. And if you see this kind of a shorthand notation, now it should be possible for you all to identify what it means. And by and large, histone methylation would silence the gene, histone acetylation would activate the gene. But of course, there are exceptions to this rule, but this is the general common rule. So having seen the mechanisms for epigenetic control, we now look at the different diseases which can happen. So over here, first is the methylation. So that means the chemical modification of the nucleotide. So this MECP2 gene, if there are mutations, and there are more than 250 mutations described for this, then there is something known as a Rett syndrome, which is a neurodevelopmental disorder, usually affecting the girls in the first year of their life, and there will be regression of their language and motor milestones. What is known as this methyl CpG binding protein 2, short form MECP2, it binds to the methylated regions of the genome and it regulates it in a way which makes it possible for normal neurodevelopment. And when there is a mutation in this particular gene, it's not able to bind to this methylated region and there is abnormal neurodevelopment. Now, neurodevelopment in utero and postnatally also it continues. And this is the proposed, we don't know exactly how this particular problem arises, but this is the current hypothesis. And in Rett syndrome, you have to remember that there's a problem in the binding of this MECP2 to the methylated region because of a mutation in this gene. The other example for us psychiatrists, this was the first time ever that a hypermethylation of the renin gene, this was published way back in 2005, was identified in the brains of patients with so-called schizophrenia and an associated increase in the DNA methyltransferase in the GABAergic cortical neurons. So in simple English, renin is a particular protein in neurodevelopment is associated with the neuronal cell migration. So if you read about the development of the brain, the neurons, they are to migrate to a particular position. And if it doesn't happen appropriately, different neurodevelopmental and neurodegenerative disorders later on can develop. And if renin is hypermethylated, it is not able to do that job. And in the adult brain, renin serves a function in synaptic plasticity. And this association with increased methylation of the GABAergic neurons. So what happens is, if you remember, methylation means the gene is switched off. And you know the GABA neurons are actually inhibitory. And loss of this inhibition, we already know happens in schizophrenia and the underlying glutamatergic overactivity. So this hypermethylation of renin was first identified in brains of schizophrenia. Other than that, the genome-wide sequencing had shown that different CGs, the cytosine guanine pairs in the frontal cortex, in this disorders, I would say psychosis, schizophrenia, bipolar, they showed extensive changes in DNA methylation of many gene networks which are involved in mitochondrial function, brain development, and stress response. But it's still not clear whether these methylation changes were a cause or a downstream effect of the pathology or involved in the progression. But what is known is this methylation was actually happening in the functional gene networks, which were identified by the different genetic studies. And definitely, this shows that there is an important role of epigenetics in the different psychiatric disorders that we are seeing every day in our clinics. The other histone modifications and the disease, that's not so common, but with increased use of the genome sequencing, more and more disorders are getting identified. The hypothesis is that if there is a histone modification, either there is redundancy, that means it can be done away and something else can work, or it can be potentially lethal. So this particular embryo or fetus never sees the light of the day, and therefore we don't see that much in our practice. But if there is a defective histone modifying enzyme, it can directly cause diseases. And two examples are shown on the slide: Beaver syndrome and brachydactyly mental retardation syndrome. I won't go into details, but there's a problem with methylation in one and histone deacetylase in the other. But what is more important over here is there can be also an indirect modification of the histone by another illness. The example shown here in this slide is on Huntington's disease. Of course, Huntington's disease is not anything to do with an epigenetic problem per se. We know that it is a primarily autosomal dominant illness inherited, and there are trinucleotide repeats of CAG, and there's a problem in the Huntington gene. There is formation of this polyglutamines in the Huntington protein, which eventually causes progressive neurodegeneration and proves
Ashutosh,
to be fatal.
maybe another 5 minutes, 7 minutes.
Sure. This is my second last slide. And these polyglutamines, by mechanism still not known, they cause a loss of histone acetylation. And what is known as in animal models, HDAC inhibitors can have some improvement. Human trials have not yet been done. So this is just an indirect way as to how histone modifications can present with a problem in Huntington's disease also. So to summarize, life on Earth evolved almost 3.5, 4 billion years back. Cell is the basic unit of life. DNA controls the cell function by coding for more than 100,000 proteins in humans, and proteins are essential for our structure and function of the cells. The proteins are manufactured by the process of transcription, processing in the nucleus, translation and post-translation modifications in the cytoplasm. Various proteins, RNA polymerase II being the chief one, but more than 50 different promoters, activators, the different co-activators, enhancers, silencers, all these are required for initiation of transcription to generate a primary transcript. And then DNA binding motifs, they help the transcription factors to identify the specific DNA sequences to be transcribed. The primary RNA transcript undergoes processing by splicing in the nucleus to give rise to the mature mRNA, which contains the exons, which is transported to the cytoplasm. And the genetic code codes for 20 different amino acids, which are the building block for polypeptides and proteins. And the mRNA binds to the transfer RNA to generate the polypeptide chain, which then generates a protein. And this protein undergoes post-translational modifications. And gene expression is all the process, right from transcription of the genetic code in the cell nucleus to translation of the RNA code and formation of proteins in the cell cytoplasm. And epigenetics is defined as a modification of the genome heritable during cell division that does not involve a change in the DNA sequence. And two chief mechanisms of epigenetic regulation are chemical modification of the individual nucleotides that make the genome and chemical modification of the histone proteins which pack this genome. And of course, a third mechanism by non-coding RNAs, which I have left out of this basic view. And DNA methylation generally suppresses the gene expression. Demethylation promotes gene expression. Examples, Rett syndrome, renin gene hypermethylation in psychosis, and histone acetylation generally promotes gene expression, and deacetylation suppresses gene expression. Examples I saw of Weaver syndrome and indirect histone modification in Huntington disease. And before I conclude, the most important slide of today's presentation is this, and I will read it out. The field of epigenetics is susceptible to overinterpretation, unrealistic hype, and even deliberate misinterpretation. So therefore, experts and the public alike need to exercise a degree of caution when it comes to claims about epigenetics. Epigenetics helps us to understand a lot between the genotype and the phenotype and the influence of environment, but it is not the panacea for everything that we know. So please be very, very cautious if you read a paper or a claim that epigenetics is the whole and soul for everything. And I have recommended one book if you all would like to start reading about epigenetics. This book, Introducing Epigenetics, would be a good starter by Ennis and Pug. And this is what would be a good starting point for all of you. That's all from my side. Over to you, sir.
Thank you. Thank you, Dr. Ashutosh. There's a very elaborate description of what has happened to nature and human race over the last 4 billion years. And you have in great details explained to us how DNA and mRNA eventually reflect on the protein synthesis in the brain and the body. I have been interested in epigenetics from a slightly different and more a clinical angle. In the sense, Why it happens is what you told us. And what happens or how can we utilize it is something that is for us to know. And I read somewhere that understanding the epigenetics helps us to identify or propose some biomarkers in the future. For a clinician, it is the biomarkers rather than the genesis of the biomarkers that would be of relevance. So, for example, you mentioned Huntington's disease and histone. So, it's possible that at some point in time, we'll be able to have some kind of a histone marker which cautions us and figure out, nowadays we do this prenatal testings for various possible genetic disorders. So I hope that at some point in time, with better understanding, there's phenomenal rise in the amount of understanding in epigenetics in the last 5 years. So we should be able to identify the biomarkers or some markers, the histone, for example, in Huntington's. And if we can do some kind of a cell typing of histones, metabolism in a person or in a pregnant woman where there is some history of inherited disease like Huntington's, we might be able to prevent the disease. I was also kind of quite interested in realizing that this catechol O methyltransferase gene is associated with some kind of general cognitive function. And a drop in the general cognitive function is associated with a phenotype eventually of schizophrenia and bipolar disorder. So I want to know whether you at this point in time with your extensive study would even conceptualize, leave alone giving an answer, conceptualize that somewhere in the future, we'll have genetic testing, prenatal genetic testing to identify possible mental illnesses like schizophrenia or depression or Huntington's disease in the progeny.
To be honest with you, I don't have the answer for your proposal, but it will be far very complex, because if you remember the whole transcription factor, 50 different proteins and different permutation combination of response elements, almost 1500 different 1800 different transcription factors. It's really a mind, I would say, numbing to even imagine what are the possible permutation combinations. So today, as more and more genome sequences are happening, there's something known as an ENCODE project. Like you had the Human Genome Project, which actually just gave the protein coding. Now they are going ahead with this epigenome also. But the day which you are kind of thinking about, I see it many centuries down the line, not
Many
at least in...
I thought less than a decade down the line, because now we also have exome sequencing, for example.
Yes, but if you remember, sir, I said that the exact causal relationship between a histone modification and the genome functioning is still not established. So actually, all this is still coming at a associational level. We are still not seeing it as a causal direct relationship, though that's possible.
Yeah.
But
Go
it's
ahead.
still not yet well established. So if there is a histone modification, or if there is a methylation of one of the cytosines, how does that change the genome functioning? Is it causal, or is it because the cell wants to express it is actually kind of acetylating the methone histones? This is still not known. So we are really at a scratch level of all this.
I was speaking to some people. I don't know whether I should name them because I don't have their permission. But they said it is important to study epigenetics simply from, looking at how science has progressed in the last few years. For example, 10 years ago, HIV was considered untreatable. And now we know it's not so because we have a proper science data. So likewise, once we have a now we know that something happens at the epigenetic level. And we know there is something called methylation. And maybe there will be biomarkers of these methylation that will help us. For example, this COMT gene, catechol O methyltransferase gene, is now, there are 7 genes which are earmarked for this kind of studies. So maybe we'll get something. I would hope we do. There's a question from Shalini Anand. She wants to know
Yes,
what is the role of epigenetics in PTSD, especially in the cases of CPTSD.
I don't know what is CPTSD. If Shalini is still around, maybe she can choose to elaborate what is CPTSD. But anyway, I'll come to the question. Complex PTSD. Okay. So let's look at it this way. This whole concept of trauma causing different disorders is not so straightforward. Why? If we have to establish the trauma as being causative. So first of all, we are looking at a trauma, and trauma can be anything: psychological, biological, physical, social. There has to be mechanisms by which this trauma is going to percolate down to the cellular level, and we are talking of in the brain, within the neurons, and then it is supposed to be causing the problem. Evidence is very weak, and lots, if you remember the last slide, there's a lot of hype and lots of over misrepresentation, I would say. These studies have a small sample size, lot of confounding variables, and more importantly, the confidence intervals are very wide. So actually, it's not a simple trauma and a particular disorder. That's not how it happens. Because there is a selection bias, sampling bias in this particular studies, even the basic, I would say, the phenotype, at the base, we don't know how much of it was contributed by the genotype and by the environment. So that itself is not known. So then how can we study about it? Secondly, even if we are talking of a trauma, that means we are talking of an environment. At any given point in time, you cannot have a homogenization of that environment. There'll be lots of multimodal stimulus coming in, and it will vary over time. So something that happened ages ago, giving rise to some finding today, 10 decades down the line, is really hard to figure out. And thirdly, if these studies have actually to be done, there is something known as exposome, and we need at least 2-generation exposome data to say that yes, trauma was heritable, etc. So in humans, it's not straightforward. At least all this nowadays, the concept of transgenerational epigenetics, intergenerational epigenetics, this is so far shown only in worms and in the fruit fly, and that too with something like modifying the temperature or giving them less oxygen, etc. Not psychological trauma. Then it comes to the mice and the mammals, the evidence is really weak. There is some possibility, but it's not as much as what is been kind of hyped about. So that is what I will like to say for now.
That's very nice explanation, Dr. Ashutosh. Shalini, I might just add here, if you permit. I sat through some discussions on PTSD, the whole concept of PTSD. When it came out after the Vietnam War, the 13% of combat exposed soldiers developed PTSD, and millions and trillions of dollars got spent on researching on PTSD. And somebody said that if 13% of combat exposures got PTSD, 87% did not. So should we be studying the epigenetics of the 87% who did not break down in response to the combat stress, which was identical in both the samples? And a psychiatrist from the US Army actually went and studied the pre-morbid personality of the PTSD as well as the new recruits into the army. And he found that the amount of pre-morbid mental status abnormalities are high in people joining certain services. So there's a lot more to PTSD than what meets the eye. Dr. Jaya Jacob wants to know, she says, can you please repeat the name of the book you suggested to read on epigenetics? And I think you already answered that,
Yes.
right? And then
Yes.
Dr. Ritvik Chatterjee says, probably it's VV, so it's meant for me. I do think that it will be possible. However, we'll have to let go of the terms like schizophrenia or anxiety or bipolar disorder. Instead, we can talk about this child is likely to have mutation, and whatever mutation is likely to have certain symptoms depending upon the lifestyle. Well, that's really, really true, Dr. Ritvik, because the diagnosis that we have just now is by cluster diagnosis. This, that, and those symptoms put together give a diagnosis. So I have been discussing this times and again that DSM and ICD are to be used as language of communication between mental health professionals rather than anything to do with the etiology of that so-called disorder. When you and I speak of, say, depression, you and I, we know that we mean a cluster of these symptoms. So let us treat DSM and ICD as language of communication rather than Bible of diagnostics. And of course, the insurance company would require it. Shivam Jaiswal is very pleased with your presentation, Dr. Ashutosh. A great presentation,
Shivam.
sir. There has been a rise in repurposing the drug targets of all existing drugs to overcome the increasing drug resistance in patients. And his question is, can we repurpose these drugs to epigenetic biomarkers and control the progression of the diseases and potentially cure them as well? You got that question, Dr. Shivam Jaiswal? The question is, can we repurpose the existing drugs to epigenetic biomarkers and control the progression of the diseases and potentially cure them as well? Ashutosh, your video has got switched off.
I switched to my phone. That is why.
Okay. All right.
So, well, I don't know the answer to the question by Dr. Shivam. Each and every molecule that we are using has an epigenetic effect. Each and every molecule that we are using. Today, it's too early and too premature to say we can just repurpose a molecule as an epigenetic biomarker or for some purpose. But please remember, valproic acid, which you are using, already has a HDAC inhibitor activity. So when you use this molecule, you're going to prevent certain genes from getting expressed, and that could help in patients of psychosis. So just understand that valproic acid, what you're using, even clozapine, for that matter, all the molecules that you're prescribing are having some or the other epigenetic action, most of which I'm not aware of, and we are barely aware of what actually is happening inside the body.
That's a good explanation. Dr. Shalini and Dr. Ritvik, I might just add here that this whole molecular and genetic and epigenetics in psychiatry is a relatively new science. It started in 1987 in the US. And then we started getting this Journal of Molecular Psychiatry and Translational Psychiatry, which are relatively recent. But if you do get an opportunity, at least even if you get access to the entire article, try and read the summaries of these, which are available on the net free. Both the Journal of Translational Psychiatry and the Journal of Molecular Psychiatry, they are published by the Nature. And it's possible. So that it's great to have this question. So both Shalini and Ritvik were answering that CPTSD was complex PTSD. Shalini says may not be a reliable finding from the logical positivistic position, but clinically huge evidence is there. And yeah, of course, there is. And unlike Dr. Ashutosh saying will take centuries for the biomarkers to emerge, I'm a little positive it may not happen in my lifetime, but it will certainly happen sooner than that. Dr. Jaya Jacob is thankful to you, Dr. Ashutosh, for a nice presentation. And I think with that, we end whatever that has appeared on the chat box. Ashutosh, thank you very much for this lovely presentation. You had worked very hard at it. I'm not sure if I understood, even if I understood 20% of what you said, it will probably make me a better psychiatrist. Thank you very much, Ashutosh. And over to you, Dr. Rashmin.
Thank you.
Thank you very much, Dr. Ashutosh. Thank you very much, Dr. Bhaiya. Absolutely brilliant is all I can say. Epigenetics for dummies is something which probably I relate to a lot more in the context of this discussion. And the beauty of the chat was how lucidly you started with right from the basics and then went on to the complexities of what epigenetics is all about. And I'm sure many people would have liked to be here, but they may have missed the opportunity. And these lectures are going to be available for free viewing anytime on the website for eternity, as long as the website is up forever. So I would invite all of you to take advantage of that opportunity and keep revisiting this lecture, because this is the most comprehensive coverage of the basics of genetics, not just epigenetics, but genetics as well. So that would be available for everybody to be able to learn from and enjoy. Thank you very much once again, both of you, for having made the time.
I speculate, Rashmin, that discussions like this is just the first installment of various other things that will come maybe once in 2 years, but because the way the science is progressing is phenomenal. And thank you for conceptualizing this and giving me an opportunity to be here today. Thank you very much.
The pleasure is all mine. And thank you all very much for being with us today and continuing to support and participate in this lecture series. And of course, thank you very much, Sun Pharma, for their unflinching support. And we would be more than happy to have your suggestions about what are the topics you would like, what are the areas you would like us to address, what speakers are of interest to you. We will try to incorporate everybody's suggestions, and we look forward to having these kind of fruitful discussions. But I think the discussion part was almost as exciting as the actual talk itself. So that tells us how invested people were, and we are encouraged by your participation. So thank you very much, all of you, once again. And I wish you a great evening ahead. Thank you.
Thank you, Rashmi. Have a great evening too.
Thank you.
Bye-bye.
Thank you very much. Thank you.
Bye, Ashutosh.
Bye.